Welcome to the Deep Dive, your shortcut to truly understanding complex biological processes. Today, we're plunging into something really fundamental, incredibly elegant, how the cells in your body, the very building blocks of life, talk to each other, even across, huge distances. It's this constant, complex cellular chat that's essential for pretty much everything. Okay, let's unpack this.
Right, and for this deep dive, we're leaning heavily on a fantastic resource, Medical Physiology by Boron and Boulpaep. We're zeroing in on signal transduction. Our aim here is to take this dense, detailed information and make it really clear, conversational, and frankly usable for you, whether you're hitting this for the first time in college or you're gearing up for med school. Because understanding how cells communicate, well, it's not just abstract theory. It underlies everything from how an embryo develops to how your body responds to, say, a simple cut or a major infection.
Exactly. Think of it like this. We're mining for the gold nuggets of knowledge, connecting the big ideas to the details that actually matter, and showing you how it all ties back to real clinical practice. And we promise, no slides, no diagrams needed. We'll explain it all from the ground up. It's your shortcut to really getting it. So let's start right at the beginning. The core idea. The big picture is surprisingly simple, right? Every single cell in your body has the ability to receive information and then, do something with it. And the main way they do this, it's through chemical messengers, a whole network of them.
That's the essence of it. And these chemical signals, they get delivered in a few main ways. Think of them as different postal services for cellular messages. First up is endocrine signaling. This is the classic hormone pathway you probably learned about. A cell, or gland, makes a chemical, releases it into the bloodstream, and it travels potentially a long way to act on distant target cells. Think insulin for the pancreas acting all over the body, or hormones from the pituitary gland.
Okay, so that's the long-distance service. What else?
Then there's paracrine signaling. Here, the signal acts only on cells right next door, in the very same tissue. Now, for this to work locally, the signal molecule can't spread too far, so it's usually rapidly grabbed by nearby cells or broken down by enzymes, or even trapped in the local environment. A really classic example is the neuromuscular junction, where a nerve tells a muscle to contract. The nerve releases acetylcholine, or AC,
Right.
which quickly diffuses across a tiny gap and activates receptors on the muscle cell. Bang, the muscle contracts. But then almost instantly, an enzyme called acetylcholinesterase chews up the ACA, stopping the signal. Very fast, very local.
So it doesn't keep telling the muscle to contract forever.
Exactly. Termination is key. And the third mode is autocrine signaling. This is where a cell releases a signal that actually binds to receptors on its own surface. It's basically the cell talking to itself. Often used for feedback control, sort of like saying, okay, I've sent the message, I can stop now.
Self-regulation. Got it. So we have these delivery methods, endocrine, paracrine, autocrine. Now let's talk about the messages themselves and the mailboxes, the receptors. What are these signals made of?
Well, they're a diverse bunch. You've got amines like epinephrine, adrenaline, peptides and proteins like insulin or growth factors, steroid hormones like estrogen or testosterone, and even really small molecules, amino acids, nucleotides, even ions like calcium or gases like nitric oxide can act as signals. And for any of these to work, they need to bind to a receptor. A receptor is typically a protein, sometimes a lipoprotein, that's specifically shaped to bind one particular signaling molecule, its ligand, like a lock and key.
And these receptors aren't all in the same place, right? You mentioned steroids earlier.
That's a good point. Most of these signaling molecules, especially the proteins and peptides, are water-soluble. They can't just pass through the cell's fatty membrane. So they bind to cell surface receptors, proteins embedded right in the plasma membrane. But those hydrophobic ones, like steroid hormones or vitamin D, they can diffuse right through the membrane. Their receptors, the intracellular receptors, are waiting for them inside the cell, either in the cytoplasm or sometimes directly in the nucleus.
Okay, so the location matters. And I imagine the type of receptor drastically changes how the signal is handled inside the cell. They must use different internal machinery.
Absolutely. We can broadly classify receptors into four major categories based on their signal transduction mechanism, basically, how they relay the message onwards. First, ligand-gated ion channels. These are super straightforward. The receptor protein is the ion channel. When the ligand binds, boom, the channel opens or closes, directly changing the flow of ions across the membrane. Think of the nicotinic acetylcholine receptor at the neuromuscular junction again. It's fast and direct.
Receptor equals channel. Simple enough. What's next?
Second, the largest family. G protein-coupled receptors, or GPCRs. These are also membrane proteins, but they work indirectly. Binding of the ligand activates the GPCR, which then activates an intermediary molecule called a G protein. It's this G protein that then goes off to interact with other downstream targets, like enzymes or ion channels.
Okay, so an extra step involving this G protein.
Exactly. Third, we have catalytic receptors. These membrane proteins either are enzymes themselves or they form a complex with enzymes. So when the ligand binds, the receptor's own enzymatic activity is switched on, or it activates its associated enzyme partner. The receptor itself does the work.
Binding triggers enzymatic action. Got it. And the fourth.
Finally, nuclear receptors. As we mentioned, these are inside the cell. They bind to ligands like steroid hormones. And importantly, these ligand-bound receptors function as transcription factors. They go directly to the DNA and regulate which genes get turned on or off. They provide a direct link between an external signal and gene expression.
Wow, okay. Four very different strategies. But despite this diversity, you said there's a kind of common flow to how these signaling events unfold, especially for the membrane receptors. Here's where it gets really interesting.
Yeah, there's a general sequence. Think of it in 6 steps. 1, recognition. The signal, the ligand, binds specifically to its receptor. It's usually weak, non-covalent binding, which is important so the signal can eventually stop. 2, transduction. Ligand binding causes the receptor to change shape. This change initiates the first intracellular event, maybe generating a second messenger or starting a phosphorylation cascade. 3, transmission. If there's a second messenger, its signal gets relayed to the ultimate effector proteins, maybe enzymes, channels, or transcription factors. 4, modulation. This often involves protein kinases adding phosphate groups and protein phosphatases removing them. Phosphorylation is like a molecular switch, turning proteins on or off. 5, response. This is the cell's overall reaction, often integrating inputs from multiple signaling pathways happening simultaneously. And 6, crucially, termination. Feedback mechanisms kick in at various points to shut down the pathway once the signal is no longer needed. You need an off switch.
Recognition, transduction, transmission, modulation, response, termination. Okay. That makes sense as a general framework. Now, besides these chemical signals flying around, you also mentioned cells can communicate through direct physical contact.
Yes, absolutely. Direct cell-to-cell interaction is vital. One way is through gap junctions. These are basically tiny tunnels, water-filled channels that directly connect the cytoplasm of adjacent cells. Small molecules and ions like calcium or KMP can pass right through. They're super important for coordinating activity in tissues like heart muscle or smooth muscle, allowing electrical signals to spread rapidly. And they can open or close quickly, for instance, if a cell gets damaged. High calcium inside it can trigger its gap junctions to close, protecting its neighbors.
Like closing a bulkhead door on a ship.
Mm-hmm.
Smart. What other kinds of contacts?
Then you have adhering junctions. These use proteins called cadherins to basically glue cells together. They link to the cell's internal skeleton, the actin filaments, providing structural integrity. But they're not just structural, they also signal. If these junctions get disrupted, say by certain growth factors, a protein called beta-catenin can break free, head to the nucleus, and turn on genes involved in cell proliferation and movement. Important in development and unfortunately also in cancer metastases.
So even the glue sends messages.
It does. And similarly, tight junctions. These form seals between cells, like the caulking between tiles, creating barriers, like in the lining of your gut to prevent leakage. But again, proteins within these junctions, like ZO1, also have signaling roles. And clinically, mutations in related proteins can even affect things like blood pressure regulation, causing some forms of hypertension.
It's amazing how structure and signaling are so intertwined.
It really is.
Yeah.
And one more type of direct contact, membrane-associated ligands. Here, the signal isn't soluble. It's actually a protein embedded in the membrane of one cell. And the receptor is on the neighboring cell. They have to physically touch for the signal to be sent. This is crucial for things like guiding nerve axons to their correct targets during development, providing spatial cues.
Okay, that covers a huge amount of ground on how signals get sent and initially received. Let's circle back to those second messengers you mentioned, like KMP or calcium. You said they act as amplifiers.
Yes, amplification is a key feature. Think about it. A single hormone molecule binding to one receptor might only last for milliseconds. But that one event can trigger the activation of, say, hundreds of G proteins. Each of those might activate an enzyme like adenyl cyclase, which can then churn out thousands of KMP molecules. So from one initial signal, you get a massive intracellular response. A tiny concentration of epinephrine, like 10 to the minus 10 molar, can rapidly boost intracellular KMP levels 10,000-fold. That's serious amplification, leading to a significant physiological change like glucose release.
Wow, so a whisper outside becomes a shout inside. And you also mentioned specificity and diversity.
Right, specificity comes from the receptor. Different signals might use the same downstream pathway, like SAMP, leading to the same cellular effect. For example, several different hormones can trigger fat breakdown via SAMP. But diversity is equally important. The same signaling molecule can cause completely different responses in different cell types because those cells have different receptors or different internal machinery linked to the receptor. Our old friend acetylcholine is a perfect example. It causes skeletal muscle to contract, but it slows heart muscle contraction and it triggers secretion in pancreatic cells. Same molecule, different outcomes based on cellular context.
Fascinating. And this allows for complex, coordinated responses.
Exactly. Think fight or flight. Epinephrine is released. In the liver, it triggers glycogen breakdown for energy. In skin blood vessels, it causes constriction. In skeletal muscle blood vessels, it causes dilation. In the heart, it increases rate and force. All these different coordinated responses prepare the whole organism for action, all initiated by one hormone binding to different receptors in different tissues. And complex cell behaviors, like deciding whether to divide, often require input from multiple signaling pathways converging what we call crosstalk.
Okay, this complexity is incredible. Let's dive deeper now into some of these specific systems. You mentioned GPCRs are a huge family. Let's start there.
Right, the G protein-coupled receptors. Huge family, characterized by that structure weaving through the membrane 7 times. They partner with these G proteins, which are heterotrimeric, meaning 3 different subunits, alpha, beta, and gamma. They're also related small GTP-binding proteins like RASA, but let's stick with the heterotrimeric ones for now. These G proteins are molecular switches. When inactive, the alpha subunit is bound to GDP. When the GPCR gets activated by a ligand, it nudges the G protein to release GDP and bind GTP instead. That GTP-bound state is the on switch.
And there are different types of G proteins.
Many different types. Different alpha, beta, and gamma subunits combine in various ways. This diversity allows different receptors to link to different downstream effectors. We often categorize them by the alpha subunit. For example, GS stimulates adenyl cyclase, increasing KMP, while G inhibits it, decreasing KMP. GQ activates phospholipase C. This is clinically so relevant. Take epinephrine again. In the heart, it binds beta-1 receptors linked to Gs, increasing KMP and heart rate. In some blood vessels, it binds alpha-2 receptors linked to G, decreasing KMP and causing constriction. It's all about the specific GPCRG protein pairing.
And this system can be hijacked, you mentioned.
Definitely. Bacterial toxins often target G proteins. Cholera toxin, for example, modifies G as alpha, so it can't turn itself off. It can't hydrolyze GDP back to GDP. So G also stays permanently on, leading to massive, uncontrolled HEMP production in intestinal cells. This causes ion channels to open, water floods out, leading to severe diarrhea, the hallmark of cholera. Pertussis toxin from whooping cough does the opposite to GI. It modifies GL-alpha so it can't be activated. It gets stuck in the off state. Some inhibitory signals can't get through, and again, you end up with too much KMP because the brake is broken.
Wow, so these toxins exploit fundamental cellular switches. Can you walk us through the normal G protein activation cycle quickly?
Sure. Step 1. Ligand binds the GPCR, activating it. Step 2. Activated GPCR bumps into an inactive G protein and promotes the swap of GDP for GTP on the alpha subunit. Step 3. The G protein now dissociates from the receptor. Step 4. The activated G protein, alpha GDP, usually splits from the beta-gamma complex. Step 5. Both the alpha-GDP and the beta-gamma complex can then interact with the respective downstream effector proteins, they're both active signalers. Step 6. The alpha subunit has an intrinsic GTPase activity. It slowly hydrolyzes GTP back to GDP. This turns itself off. The inactive alpha-GDP then reassociates with beta-gamma, ready for another cycle. There are also proteins called RGS proteins that help speed up this off switch. Okay, so both alpha-GTP and beta-gamma can carry the signal forward. What kinds of things do they actually do? The alpha subunits have diverse targets. As we said, GS-alpha activates adenyl cyclase, G-alpha inhibits it. GQ-alpha activates phospholipase C, PLC. G-alpha, transducin, found in the eye, activates a phosphatase that breaks down cGMP. Some G-alphas can even directly interact with and modulate ion channels.
And the beta-gamma subunits, they're not just passive partners.
Not at all. The beta-gamma complex is a signaling molecule in its own right. A great example, acetylcholine released by the vagus nerve slows the heart. It binds to a muscarinic GPCR. This releases a beta-gamma complex, which directly binds to and opens potassium channels in the heart muscle cell membrane. This makes the cell hyperpolarize, harder to excite, thus slowing the heart rate. Beta-gamma can also modulate adenylyl cyclase, PLC, and activate another enzyme called phospholipase A2. And they play a role in turning the signal off too, by recruiting kinases that phosphorylate the receptor itself, marking it for removal, a process called desensitization.
Okay, that covers the big G proteins. What about those small GTP binding proteins you mentioned, like REST?
Right, the small GTPases. These are single subunit proteins, but they work on the same GDP-GDPP switch principle. There are several families: ROS, RO, RAB, ARF, RAN, each involved in different things. The ROS family is critical for relaying signals from receptor tyrosine kinases, we'll get to those, to the nucleus, ultimately controlling gene expression and cell growth. And clinically, this is huge. Mutations in ROS genes that lock ROS in the active GTP-bound state are found in a large percentage of human cancers. These mutated ROS proteins constantly tell the cell to grow and divide. They become oncogenes. Other families control different jobs. RO manages the actin cytoskeleton, cell shape and movement. ROB and ARF handle vesicle trafficking. They're involved in almost everything. And like the big G proteins, they have helper proteins. GEPs speed up inactivation, GES promote activation.
So many switches. Let's focus now on the signals produced by these pathways. You mentioned CMP and cGMP, the cyclic nucleotides. Let's dive deeper there.
Okay, CMP, generated by adenylyl cyclase, usually activated by GS-coupled receptors. Its intracellular concentration rises rapidly. What it does then depends entirely on the cell type. In the adrenal cortex, ACTH stimulates cortisol release via CMP. In the kidney tubules, vasopressin increases water reabsorption via KMP. We already mentioned the pathological site cholera, McGune-Albright syndrome, where excess KMP causes havoc. The main way KMP exerts its effects is by activating KMP-dependent protein kinase A, or PKA. PKA is normally inactive, a complex of regulatory and catalytic subunits. When KMP levels rise, CMP binds to the regulatory subunits, causing them to release the active catalytic subunits. These active kinases then go and phosphorylate specific target proteins, channels, enzymes, other signaling proteins, changing their activity. PKA can even travel to the nucleus to phosphorylate transcription factors and change gene expression.
And how does the cell control where PK acts?
Cleverly, using a kinase anchoring proteins, or AKAs. These proteins bind to PKA and tether it to specific locations within the cell near a particular ion channel or at the nucleus, ensuring KMP signals are localized and efficient. We also mentioned amplification, one hormone molecule leading to thousands of KMP molecules, which activate many PKA molecules, which can phosphorylate countless target proteins. Huge signal boost.
And how is this KMP signal turned off?
Two main ways. First, enzymes called phosphodiesterase, PDEs, constantly break down KMP into plain AMP, lowering its concentration. Caffeine, incidentally, inhibits some PDEs, which is partly why it has its stimulant effects. Second, and just as important, are protein phosphatases. These enzymes do the opposite of kinases. They remove the phosphate groups that PKA and other kinases added. There are several families like PP1, PP2A, PP2, also called calcineurin. Calcineurin is calcium activated and is the target for immunosuppressant drugs like cyclosporine and tacrolimus used after organ transplants. Removing the phosphates resets the system.
Okay. And briefly, cGMP.
cGMP is similar but used in different pathways. We mentioned its role in vision, where light leads to cGMP breakdown, closing channels. It's also produced by guanylate cyclases, like the ones activated by nitric oxide, or ANP, often leading to smooth muscle relaxation.
Got it. Cyclic nucleotides, CMP and cGMP, controlled by cyclases, PDEs, kinases, and phosphatases. Now, what about that other major branch of second messengers you mentioned earlier, the ones derived from membrane lipids?
Right, the phosphoinositide pathway. This starts when a receptor, often coupled to a GQ protein, activates phospholipase C, PLC. PLC's job is to cleave a specific membrane phospholipid called PIP2. This cleavage yields two distinct second messengers: IP3, inositol 1004-5 trisphosphate, and DAG, diacylglycerol.
Two messengers from one reaction. What do they do?
IP3 is small and water-soluble. It rapidly diffuses through the cytoplasm to the endoplasmic reticulum, ER, which is a major storage site for calcium inside the cell. On the ER membrane, IP3 binds to the IP3 receptor, which is itself a ligand-gated calcium channel. Binding opens the channel, and whoosh, calcium ions flood out of the ER into the cytoplasm. This causes a sharp, rapid rise in intracellular free calcium concentration. And calcium is a hugely important second messenger in its own right.
What kinds of things does this calcium spike trigger?
Oh, a vast array. Muscle contraction, release of neurotransmitters, secretion of hormones like insulin, changes in enzyme activity, even gene expression. It's incredibly versatile. The cell keeps resting calcium levels very, very low, so even a small influx represents a big relative change, a strong signal. A key mediator of calcium's effects is a protein called cammodulin. CAMLM. When calcium binds to calmodulin changes shape and can then bind to and activate a whole range of other proteins, including CAM kinases, calcium calmodulin-dependent protein kinases, which then phosphorylate their own targets.
So IP3 leads to calcium release, which activates calmodulin and kinases. What about the other product, DAG?
DAG, being lipid-like, stays embedded in the plasma membrane. Its main job is to recruit and activate another important kinase, protein kinase C, PKC. There are different types of PKC, some requiring both DAG and the high calcium levels triggered by IP3 for full activation. Activated PKC then phosphorylates its own set of target proteins, influencing things like cell growth, membrane structure, and gene expression. Interestingly, some potent tumor promoters found in nature, like 4-ball esters, work by mimicking DAG and inappropriately activating PKC, driving uncontrolled cell proliferation.
So PLC activation gives you two signals, IP3 driving calcium release and DAG activating PKC, often working together.
Exactly. And the cell has mechanisms to turn this off too. IP3 is quickly broken down and calcium is pumped back into the ER or out of the cell by ATP-dependent pumps.
Okay, that's a complex system. Are there other important lipid-derived signals we should know about?
Yes, definitely. We need to talk about arachidonic acid, AA, and its metabolites, the eicosanoids. AA is a 20-carbon fatty acid that's usually stored in membrane phospholipids. It gets released by the action of phospholipase A2, PLA2, often triggered by other signaling events. Once free, AA doesn't usually act directly. Instead, it's rapidly converted into a whole family of potent, short-lived local hormones, the eicosanoids. There are three main enzymatic pathways for this.
3 pathways from one fatty acid. Okay, what are they?
First, the cyclooxygenase, COX pathway. There are two main COX enzymes, KOX1 and KOX2. They convert AA into intermediates that lead to prostaglandins, prostacyclins, and thromboxanes. These have really diverse, often opposing effects. For example, thromboxane A2, TXA2, mainly made by platelets, promotes platelet aggregation and constricts blood vessels, helps form clots. Prostacyclin, PGI2, made by endothelial cells lining blood vessels, does the opposite, inhibits platelet aggregation and dilates blood vessels. It's a delicate balance.
Ah, and this is where aspirin comes in, right?
Exactly. Aspirin irreversibly inhibits COX enzymes, particularly COX1 at low doses. By blocking TXA2 production in platelets, it reduces clot formation. That's why it's used to prevent heart attacks and strokes. The downside is that COX1 also makes protective prostaglandins in the stomach lining. Inhibiting it can lead to stomach ulcers and bleeding, a common side effect of aspirin and other traditional NSAIDs, non-steroidal anti-inflammatory drugs, like ibuprofen, which also block COX enzymes. KOX2 is usually induced during inflammation and contributes to pain and swelling. So selective COX2 inhibitors like celecoxib, Celebrex, were developed to reduce inflammation with hopefully fewer stomach problems, though some concerns emerged about potential cardiovascular risks with some COX2 inhibitors.
Okay, so that's the COX pathway. What's the second pathway for AA?
The 5-lipoxygenase, LOX pathway. This enzyme converts AA into leukotrienes. Leukotrienes are major players in inflammation and allergic responses. Some attract immune cells, while others, particularly the cystinoid leukotrienes, are extremely potent constrictors of airway smooth muscle, much more potent than histamine. They're heavily implicated in asthma. So drugs like montelukast, Singulair, work by blocking leukotriene receptors, helping to manage asthma and allergic rhinitis.
And the third pathway?
The third involves epoxidase enzymes, often part of the cytochrome P450 system. These convert AA into other molecules like HATs and ETs. Their roles are still being fully worked out, but they seem involved in regulating ion channels, blood vessel tone, cell proliferation, and inflammation, possibly playing roles in things like hypertension and atherosclerosis. Like other signals, these eicosanoids are rapidly inactivated by specific enzymes to keep their actions local and short-lived.
And you mentioned one other lipid signal.
Oh, right. Briefly, platelet activating factor, PAF. It's structurally different from eicosanoids, but is another potent lipid mediator involved in platelet aggregation, inflammation, and allergic reactions. It works through its own GPCR.
Incredible diversity just from membrane lipids. Okay, let's switch gears now to those receptors that are actually enzymes themselves, the catalytic receptors.
Right. These receptors cut out the middleman, in a sense. When the ligand binds, the receptor's own enzymatic domain gets activated. We can group them into about 5 classes. First, receptor gonadal-cyclases. We touched on these. Some are membrane-bound, like the receptors for AMP and BMP hormones released by the heart when it's stretched, like in heart failure. Binding of AMP-BMP activates the receptor's intrinsic gonadal-cyclase domain, making cGMP. This cGMP signal promotes salt and water excretion by the kidneys and dilates blood vessels, helping to lower blood pressure and reduce the heart's workload. There's also the soluble guanylyl cyclase, SGC, inside the cell, which is the receptor for nitric oxide, NO. NO is a fascinating signaling gas produced by endothelial cells. It diffuses into adjacent smooth muscle cells, binds SGC, boosts cGMP, causing muscle relaxation and vasodilation.
That's the pathway nitroglycerin uses for angina, right?
Precisely. Nitroglycerin releases NO. This NO signaling is crucial for blood flow regulation, neurotransmission, and even immune responses. A Nobel Prize was awarded for figuring this out.
Okay, so guanylyl cyclases. What's the next?
Receptor serine kinases. Here, the receptor itself has kinase activity that phosphorylates serine or threonine residues on target proteins. The main example is the receptor for transforming growth factor beta, TGF-way. The TGF family regulates tons of stuff: cell growth, differentiation, wound healing, immune responses. Usually, the ligand binds one receptor subunit, which then recruits and phosphorylates another subunit, activating its kinase domain to signal downstream.
And the third class, you mentioned these earlier.
Yes, receptor tyrosine kinases, RTKs. These are hugely important. They have intrinsic tyrosine kinase activity. Receptors for many growth factors like EGF, PDGF, VEGF, and for insulin fall into this category. Typically, ligand binding causes two receptor molecules to pair up, off. This allows them to phosphorylate each other on specific tyrosine residues within their cytoplasmic tails, a process called autophosphorylation. These newly phosphorylated tyrosines, PY sites, then act as docking platforms. They recruit specific intracellular signaling proteins that contain special binding domains, like SH2 domains, SRC homology 2 domains. They specifically recognize and bind to those phosphatyrosine motifs. This brings signaling proteins to the activated receptor at the membrane. One key protein recruited this way is GRB2, which then recruits another called SOS. SOS is a GEF4 ROS, that's small g protein we talked about. So RDK activation, SOS activation, we got ROS activation, and ROS then kicks off that whole MPK cascade. ROS activates ROF, and MAPK, ROF activates ME activates MERK, like uricary. MAPK then phosphorylates various targets, including transcription factors in the nucleus, changing gene expression to promote growth, differentiation, etc. It's a central pathway.
Wow, a direct link from a growth factor outside to gene changes inside via ROS and MEK.
It's a major engine for cell proliferation. The fourth class is tyrosine kinase-associated receptors. These receptors lack their own kinase domain, but they tightly associate with separate, non-receptor tyrosine kinases, like members of the Ciersac family or the Jake family. Many cytokine receptors work this way: receptors for interleukins, interferons, growth hormone, erythropoietin, EPO. Ligand binding brings the receptor chains together, which activates the associated Jake kinases. The Jakes then phosphorylate the receptor tails, creating docking sites for other signaling proteins, most notably the STATs, signal transducers and activators of transcription. Jakes phosphorylate STATs. The phosphorylated STATs then pair up, move to the nucleus, and directly activate gene transcription. This JAK-STAT pathway is crucial for immune responses, inflammation, and blood cell development. Different cytokines activate different STATs, leading to specific gene programs.
So JAK-STAT is another pathway straight to the nucleus.
Yes. And like RAS, mutations in components of these pathways, like kinases or JAKs, can lead to them being constantly active, contributing to cancers, especially leukemias and lymphomas. And the fifth class is sort of the opposite: receptor tyrosine phosphatases, PTPs. These transmembrane receptors have intrinsic phosphatase activity. They remove phosphate groups from tyrosine residues. An example is CD45, found on lymphocytes. It actually activates immune signaling by phosphorylating inhibitory sites on CRC family kinases. So phosphatases are just as critical as kinases for regulation.
Kinases add, phosphatases remove, a constant balancing act. Okay, one final major category: the signaling that goes directly into the nuclear receptors.
Right. These handle signals from molecules that can easily cross the cell membrane. Steroid hormones like cortisol, aldosterone, estrogen, testosterone, thyroid hormones, vitamin D, and retinoids, vitamin A derivatives. These ligands diffuse into the cell and bind to their specific nuclear receptors, which might be waiting in the cytoplasm or already be in the nucleus. The key thing is, the ligand receptor complex is an activated transcription factor.
So binding the hormone turns the receptor itself into a gene regulator.
Precisely. These receptors have distinct domains. A highly conserved DNA binding domain, often featuring zinc finger structures, allows them to recognize and bind specific DNA sequences called hormone response elements, HREs, located near the genes they regulate. They also have a ligand-binding domain and domains involved in activating or repressing transcription. So the activated receptor binds to its HRE on the DNA and recruits other proteins, co-activators or co-repressors, to either enhance or block the transcription of that target gene. This directly changes the protein production profile of the cell.
And this explains why these hormones have such widespread and often long-lasting effects.
Exactly. They're directly altering the cell's genetic program. And there's complexity here too. Different receptors can sometimes bind to the same HRE or form pairs, homodimers or heterodimers, that recognize different DNA sequences, allowing for fine-tuned and combinatorial control. It also allows for crosstalk. For example, the glucocorticoid receptor can interfere with other transcription factors involved in inflammation, which is part of how steroids suppress the immune response. And again, the connection to cancer. Mutated forms of some nuclear receptors, like the thyroid hormone receptor, related to the V or B oncogene, can contribute to malignancy.
We have covered an absolutely staggering amount of information today. From neurotransmitters acting in milliseconds across the synapse using ligand-gated channels, all the way to steroid hormones slowly changing gene expression over hours or days via nuclear receptors. It truly demonstrates the incredible complexity and, really, the elegance of how cells communicate. Signal transduction is fundamental. So what does this all mean?
Well, it means that virtually every aspect of physiology and medicine touches upon these pathways. Understanding how signals are normally sent and received and how they go wrong in disease is critical. When you're diagnosing a condition, understanding its pathology or thinking about treatment, whether it's cancer, diabetes, heart disease, inflammatory disorders, infectious diseases like cholera, you're often dealing with disruptions in these signaling cascades. And modern pharmacology is increasingly about developing drugs that specifically target components of these pathways, blocking an overactive receptor, inhibiting a rogue kinase, restoring a missing signal. Knowing the steps is essential for designing those targeted therapies.
It really is the language of the cell. And thinking about it, consider how one single type of molecule, like epinephrine, binding to slightly different receptors, can orchestrate such different yet perfectly coordinated responses throughout the body, the fight or flight response. How could we maybe harness that inherent intelligence, that specificity within cellular communication, to create even smarter, more precise medical interventions in the future? Something to think about.
Absolutely. And for all of you listening, especially students navigating this material, remember that by grappling with these concepts, GPCRs, kinases, second messengers, nuclear receptors, you are genuinely learning the fundamental language of life. Every pathway you understand builds a more solid foundation for your future in science or medicine. It can seem overwhelming, but keep at it. You're part of the Deep Dive family now, and you absolutely have what it takes to master this.